Now let's have a quick look at the types of neurons and their appearance in the basal ganglia. And we'll start with the striatum. The most common neuron in the striatum is the spiny projection neuron. And there's a photo of it of a neuron of that type on the left and a drawing on the right. And this neuron is GABAergic. And this enlargement shows lots of spines extending out from the trunk of one dendrite. And remember that spines probably have the role of allowing changes to the weight of one synapse independent of all the others. Now these neurons only fire in response to strong input. And when they fire, they produce trains of spikes lasting from 100 milliseconds up to 3 seconds, and then they become silent again. This shows a second type of neuron in the striatum, the inhibitory interneuron, which also uses GABA. And this shows a third type, the leptodendritic neuron, which probably uses the neurotransmitter acetylcholine. And this shows the spidery or giant aspiny neurons. These are definitely cholinergic, and they fire tonically, a few spikes per second. They don't burst fire, but they briefly stop firing under specific behavioral circumstances, and one of these circumstances is when rewards for behaviors are appropriate. Moving on to neurons in the globus pallidus, both segments have similar neurons. 3 main types of neuron have been observed, at least in the rat. The most common is the GABAergic projection neuron, and there are about the same number of GABAergic projection neurons in the globus pallidus as there are in the striatum. And there are also GABAergic interneurons. And in addition, there are a few cholinergic projection neurons that actually project to the cortex. About 1% of the neurons in the rat globus pallidus are this type, although it's not certain that they're present at all in the human globus pallidus. And these are drawings of neurons in the primate globus pallidus. Finally, taking a look at the neurons in the substantia nigra pars compacta. The projection neurons are dopaminergic and are not spiny, and they're also GABAergic interneurons. An interesting functional property of these dopaminergic neurons is that they fire in 2 different modes. One mode generates spikes at about 4 Hz. The dopaminergic neuron population tends to fire constantly at this rate. Some types of inhibition can reduce the proportion of dopaminergic neurons firing in this mode without changing the rate of firing for the ones that are still active. The second firing mode is that with appropriate stimulation, a dopaminergic neuron can produce a burst of spikes, with between 3 and 20 spikes inside a burst, and the inburst frequency is about 20 Hz. One major target of the dopaminergic neurons is the striatum. In the striatum, many dopamine receptors are distributed on dendritic shafts and spines away from the locations of dopamine synapses. For these receptors, the action of dopamine must occur through diffusion out of dopamine synapses into the extracellular environment. And in the striatum, a background extracellular concentration of dopamine certainly exists, and I've conceptually illustrated it by the purple dots. This background concentration is created by the tonic firing of the dopaminergic neurons. Dopamine released at synapses by this tonic firings, it leaks into the general extracellular environment. And it's this background concentration that regulates the relative activity of the D1 and the D2 populations in the striatum. Now, interestingly, the burst firing of the dopaminergic neurons doesn't affect the background concentration as much as you might expect. And that's because when a burst arrives at a synapse, it also triggers higher activity by enzymes that remove dopamine from the synaptic cleft. So a relatively small proportion of that dopamine actually leaks out into the extracellular environment. Burst firing and tonic firing are separately regulated. Blocking GABA A receptors in the SNC triggers burst firing, and blocking GABA B receptors increases the probability that a dopaminergic neuron will be in the tonic firing mode. The proportion of dopaminergic neurons in the tonic mode determines the background dopamine concentration in the striatum. Exactly how this works isn't fully understood, but for example, it is known that some important factors in the regulation of dopaminergic neuron activity are GABAergic neurons in the SNR and GPI. It could be that GABAergic neurons in the SNR target the dopaminergic neurons directly via GABA A receptors, regulating burst firing. The GABAergic neurons in the GPI target GABA B receptors on SNC interneurons that in turn target dopaminergic neurons, regulating the background concentration. But these are not the only factors influencing dopaminergic neuron activity. As discussed earlier, striatal projection neurons in the striosomes, or patches, also target the SNC and seem able to trigger burst firing, perhaps again via interneurons. The STN also targets the SNC and can have both excitatory and inhibitory effects on the dopaminergic neurons, probably also involving SNC interneurons. And finally, cholinergic neurons in a structure called the pendoculopontine nucleus, the PPN, also target the dopaminergic neurons. The PPN gets inputs from the GPI SNR and also the STN. Cholinergic inputs tend to increase the probability of tonal firing and have less effect on burst firing. So there's a fairly complex overall regulation of dopaminergic firing, with separate regulation of tonic and burst firing. In fact, even when dopaminergic neurons die off in the SNC, as in Parkinson's disease, it's observed that the level of dopamine in the striatum is maintained at normal levels until a very high proportion of the dopaminergic neurons have disappeared. The subthalamic nucleus is the only source of excitatory glutamatergic neurons in the basal ganglia. There are actually a couple of types of glutamatergic projection neurons, both of which are spiny, and once again there are also GABAergic interneurons. The glutamatergic neurons in the STN produce a steady tonal output in the absence of stimulation, and this tonal output is reduced by GPE activity. However, the STN also gets excitatory inputs from both the cortex and the thalamus, and in addition, it influences the dopaminergic neurons in the SNC.